Hello.
I'm Diana, one of the researchers at the Centenary Institute, and I'll be taking you through a day in the life of a scientist. I chose to go into medical research to work on problems that had real impacts on people's lives. I thought it was something important that I could be a part of and contribute to, but I really love it. The first part of any science project is going through what is already known, going through the literature, finding out what people have discovered and looked into, things that need further investigation, and that's where things begin, when you find the questions and the important areas, and then you think, okay, that's what we're going to look at. Next is planning. So much planning, making sure the dates line up, the timelines are looking good. But then there's the fun part, actually getting into the lab and getting to do the experiments. Of course, with a lot of help from the best RA ever, Trixie. Today's experiment is a comparison between diseased and healthy lung tissue samples. So I've got everything all set up here, liquid nitrogen to keep things at the right temperature, and here are our samples ready to go. In order to perform some staining and look at all of the different cell subtypes and see what differences there are. And here are our flow antibodies for staining. Once all of that is done, we can read our samples on the flow cytometer, and that's how we can figure out what kind of cell populations we're looking at here. Finally, it's time to analyze all of the data that the experiment gave us. And we find out what really happened in order to answer our questions and get our results. And that's that. Thanks for joining me on a day in the life of a scientist.